Hey folks, in the last video I walked you through how to use a Punnett square and why to use a Punnett square and didn't do a great job of it. But in reality, in this video, what I'm going to do is walk you through why you shouldn't use a Punnett square in certain cases. And this is when we're talking about more than one gene at a time. The last video we were talking about like fur texture or something like that. So let's do something similar. But now we're going to talk about two genes, two characteristics, specifically fur color and bone growth, as you can see. And for each one, to simplify matters, I'm going to give you two and only two alleles. Remember, in reality, there are dozens or hundreds of alleles for most genes. But in this case, we're going to talk about fur color being either brown or gray, which in this case, brown is going to be dominant, and bone growth either having a typical growth pattern or achondroplasia, which is a type of growth where the bones don't actually grow to typical size. This is the most common type of dwarfism amongst humans. So these are going to be our two varieties here in terms of bone growth. Now, the cross I'm going to give you is going to include alleles for both genes. So for the female parent here, you can see that they are homozygous dominant for the fur color and heterozygous for the bone growth. And here's the male parent. Now, what we're going to do is walk you through, the Punnett square for this. And just like last time, here's the cell for one of the parents. So this is going to be the dad, I suppose, assuming it's this male parent. So we got heterozygous for the B gene, heterozygous for the A gene. Now, I'm laying it out as if both genes are on different chromosomes. So we'll put the B gene on the larger chromosome, the A gene on the smaller chromosome, and here's our setup. Again, I'm just making this up. This is just an example to work with. As we saw last time, if you're going to go through meiosis and produce sperm or eggs, you're going to have to replicate all those chromosomes. So here we go. And then we're going to separate twice. Remember, meiosis 1 and 2. And it's going to be random which homolog goes in which direction, which means there are actually 4 different possible genotypes of sperm or eggs we could make from this parent. In this case, we're making sperm, and I'm going to draw out all the combos. So you could get the capital B, the big B chromosome of the large ones, and then for the small chromosome, you could also get the large A from that one. Now, again, you're going to get one of each chromosome size or type. You're not going to get 2 large chromosomes. You're not going to get 2 small chromosomes. That's why this Punnett square is a little bit tricky to make. A lot of folks will, in this box or in this location, put capital B, little b, or capital A, little a, and that ain't going to happen. You're going to get one of each chromosome size, one of the larges, one of the smalls, and so you're going to get one allele for each gene. This is a haploid gamete. So these are the 4 possible combinations. The other parent, in this case, only has capital B's, doesn't have any little b's. So there are actually only 2 possibilities here. For the long chromosome, they can only have a capital B. For the short chromosome, the gene on that chromosome could be either a capital A or a little a here. Now, we've set up the Punnett square. In this case, it's not even a square, right? So I think we tend to think of Punnett squares as always being squares because that's what you start with in middle school. These could be like any sort of rectangle you want. It's just a probability chart. In this case, there are only 2 possible gametes from mom. So if we actually fill this all out, I can put in all the combos together, and I'm just going to skip through these really quickly. What I want to do now is just write out all the probabilities of the different phenotypes. Now, you might remember or be able to figure out right now, just looking to the left, there are 4 possible combinations. The offspring could have brown fur with achondroplasia, brown with typical growth, etc., as you can see. The question now is, what are the percentages, or more honestly, the probabilities? So what's the probability of having an offspring that has brown fur and has achondroplasic bone development? If you look down at the Punnett square or the Punnett rectangle, 6 of these offspring, 6 of these 8 possibilities here do have a genotype with brown fur and achondroplasic dwarfism. And so I can write that in here. And the remaining two both have, again, brown fur and achondroplasia. So that leaves 0 for the other two possibilities. Okay, so you can do this. I think this is doable. I think, however, that the likelihood of you doing this properly is maybe a little bit low. It's pretty tricky to set these things up without making a big error. What I think most of you are more familiar with is the single gene Punnett squares. And I think leaning into that is, in many cases, the better option. So I'm going to walk you through how to do this using a little bit of very simple math. And I think it is a better option for most. So here's again the probabilities. We've already calculated these. I'm going to walk you through how to use multiplication. And so what we're going to do, instead of setting up this giant 2x4 Punnett square, I just want you to do what you've done before, which is set up a simple 2x2 for each of the two genes independently. So imagine that I've provided you a test question, and hopefully it would be a better question than this, but which asked you for the probability of one of the kids having brown fur and being achondroplasic. So you can see that written in orange. If we do the two Punnett squares really quickly, and I know that most of you can do these pretty quickly at this point, if you write them out, what I want you to do is now calculate the probabilities of each of those characteristics separately. So what's the probability of having brown fur, of the kids having brown fur? And if you look at that B Punnett square, all of the kids will have brown fur. In terms of their phenotype, all of them should have brown fur. If you look at the other Punnett square, half the kids will be heterozygous. One will be homozygous dominant, which means 3 out of 4 in all likelihood. So 75% probability each kid will have achondroplasia. Okay, so how do you calculate at this point the likelihood of having brown fur and achondroplasia? This is the new thing that I haven't mentioned before. Just multiply the probabilities together. That is 1 times 0.75, or 1 times 3 out of 4. It's just 75%. You multiply the two things together, and you're going to get your result. The two characteristics are calculated separately, and then you multiply them to get the probability of both of them being true. All right, you'll see that this number matches what we already have on the upper right. It's just a different way of doing the same question. All right, let's do another one. Let's imagine I ask you for the probability of having a gray-furred offspring with achondroplasia. Same thing, you can use the same two quick Punnett squares. You'll see very quickly that the likelihood of having gray fur is 0. You don't even have to look at the other Punnett square because you're at 0. Here's a third example. If you're looking for the probability of brown and typical growth, again, on the left, you can see all 4 have brown fur. And on the right, there's only a 25% likelihood of having typical bone development. All right, hopefully that helped you walk your way through the probabilities. Keep in mind, you're going to take 2 probabilities. If you're going to try to figure out the likelihood of both, you multiply them together, and that gets you your final answer, which does match what you'd get from one of those larger Punnett squares. You choose what works for you, and I hope it works out well for you. Best of luck, everybody. Talk to you next time.